Good afternoon. Thank you for the invitation, Dr. Burton, Dr. Perrin. I appreciate it, and I'm excited to share these results with you. So I wanted to start out in the first slide, just basically pointing out the well-characterized but sometimes underappreciated, although not by this group, fact that antibodies are bifunctional molecules. They contain the Fab domain, which mediates antigen binding and the specificity, and in the context of infection can block virus and target interactions and cross-link cell surface molecules. But in addition, they contain an Fc domain, which is the constant region, mediates the effector functions through interactions with Fc receptors. And so Fcs interact with two main flavors of Fc receptors, the type 1 and the type 2 Fc receptors. So type 1 receptors are members of the Ig superfamily, and they contain the classical IgG Fc receptors. By contrast, the type 2 Fc receptors are C-type lectins, and what regulates the differential interaction between type 1 and type 2 Fc receptors is the glycosylation state of the IgG Fc. So for example, an acylated IgG Fc adopts an open conformation which interacts preferentially with type 1 Fc receptors, precludes bindings with type 2 Fc receptors. On the other hand, an acylated Fc domain preferentially interacts with type 2 Fc receptors because it adopts a more flexible closed conformation and therefore precludes binding with the type 1 Fc receptors. Today we'll focus on the type 1 Fc receptors, the classical Fcγ receptors, and they also come in two different flavors, as you're well aware, the activating and inhibitory Fc receptors. They transduce different signals, the activating and inhibitory, by the nature of their ITAM or ITIM motifs in the intracellular domain, and it's the overall balance of activating versus inhibitory Fc receptor signaling which dictates the outcome of engagement of an immune complex or an antibody opsonized cell, and this is dictated by the relative engagement of activating versus inhibitory Fc receptors by the Fc domain. And so, for example, in the mouse IgG subclass system, there's actually selective Fc receptor engagement. So, for example, an IgG2A isotype antibody preferentially interacts with FcγR4, the main activating Fc receptor in mice, and has a very low affinity for the inhibitory receptor. And that gives it a very high activating to inhibitory Fc receptor ratio. By contrast, mouse IgG1 antibodies preferentially interact with the inhibitory Fc receptor and don't engage at all with the main activating Fc receptor in mice, giving it a very low activating to inhibitory Fc receptor. And this dictates the in vivo outcome of an antibody response, whereas mouse IgG2 antibodies preferentially interact with activating FcRs and mediate pro-inflammatory activities, whereas mouse IgG1 antibodies preferentially interact with inhibitory Fc receptors and mediate anti-inflammatory activities. And this is best characterized in a paper that now was about 10 years ago from Falk-Nirion, showing that in the TA99 tumor model, where you can get clearance of lung metastases of these B16 melanomas, if you adoptively transfer antibodies specific for the TA99 epitope of GP75. And so you see, if you give a T99 antibody to melanoma-bearing mice with an IgG1 isotype, you don't get any tumor clearance. And that's because of the low activating to inhibitory ratio. But if you give an IgG2A antibody with a very high activating to inhibitory ratio, you get full clearance of the tumors from those lung metastases. And I just want to make the point that there's actually dramatic differences between the mouse Fc receptor system and the human Fc receptor system that we'll come back to later in the talk. But for now, I just want to say that while there are differences in number of Fc receptors, there's also differences in the amino acid sequence of these Fc receptors and big differences in the expression patterns of the human Fc receptors compared to the mouse Fc receptors. So they're very different systems. Two other points I'd like to make about the Fc receptor system is that antibodies can link, and FcRs can link, the innate and adaptive immune system. And so what I mean by this is I'll just show the classical example, which the major mechanism of action of antibody-mediated killing of malignant cells in vivo is through ADCC, antibody-dependent cytotoxicity, or phagocytosis. And so this seminal experiment done now 15 years ago in the lab shows that if you give Rituxan to xenografted mice expressing human CD20 positive tumors, you can see the wild-type mice that get PBS showed great tumor growth, but mice receiving Rituxan don't show any tumor growth. But if you do this in Fcγ receptor deficient mice, you see that the Rituxan has no effect, showing that it is Fc receptors that mediate the in vivo cytotoxicity of IgG1 antibodies. As well, I wanted to make the point that antibodies and FCRs link the humoral and cellular immune systems. And so what do I mean by this? As discussed earlier briefly, dendritic cells primed with immune complexes more potently stimulate T cell responses than dendritic cells primed with antigen alone. And further, immune complexes are able to stimulate cross-presentation by dendritic cells, whereas plain old antigen is unable to do this. And what is the mechanism of this is that it's the FCR-dependent maturation of dendritic cells once they are bound by immune complexes through Fc receptors. You get upregulation of class 2, CD8, 86, and CD40. And when you get these mature dendritic cells, they're much more potent antigen-presenting cells. And I think the seminal experiment demonstrating this was done, again, almost 15 years ago, in which immature dendritic cells of the bone marrow of mice were primed with ova immune complexes and thereby matured, and then used to adoptively transfer into black 6 mice. And then 2 weeks later, you could challenge these black 6 mice with the B16 ova cell line and look at tumor clearance. And what you see is if you have naive mice, they all die upon the B16 ova challenge. If you have mice that were primed with mature wild-type dendritic cells primed with ova immune complexes, you see an increase in survival. But if you do this with Fcγ receptor 2b deficient dendritic cells, so whereby you've skewed the activating to inhibitory ratio in much more towards an activating component, and you can see that you get almost full survival in these mice. So you can really balance, modulate the engagement of dendritic cell FCRs with these immune complexes and drive CD8 T cell responses in vivo. And so, as I mentioned, cytotoxic anti-tumor antibodies directly kill tumor cells through ADCC, and this is a rapid and short-acting process. However, in the literature, it's been shown that the response to a single dose of CD20 antibody results in a long-term anti-tumor response in some patients. And in addition, anti-RIIA type T cell responses have been reported after rituximab therapy in some patients. And also, anti-HER2 and anti-MUC1 T cell responses have also been described in following HER2 and MUC1 antibody therapy in mice and patients. And so what this has prompted is this hypothesis of a vaccinal effect, in which a long-term anti-tumor cellular immune response against tumor cells is initiated after antibody treatment. And so we wanted to investigate this and to understand whether this happens in vivo and how it occurs in vivo. And we hypothesized that immune complexes generating during ADCC of tumor cells engages Fc receptors on APCs to prime a long-term anti-tumor cellular immune response. And so to understand this vaccinal effect in vivo, we set up an in vivo anti-CD20 tumor model based in part on work that Jean-Luc Telliud had done several years ago. And so what model we did was we injected mice with 500,000 syngeneic EL4 cells that express human CD20 as a tumor-specific antigen. On day 1, 4, 7, 11, and 14, we gave mice 100 micrograms of an anti-human CD20 antibody with the IgG2A isotype. So this is the potent activating receptor engaging antibody in mice. So this is the cytotoxic isotype in mice. And what you can see is, of course, if you give mice just PBS, they all die after about 2 months. But if you give mice the anti-human CD20 antibody with the IgG2A isotype, you get complete survival. And if you do this same experiment in mice lacking Fc receptors, as you would expect, you see complete killing of the mice within about 40 to 50 days, indicating that the initial clearance of tumor cells by CD20 antibody in vivo is Fc receptor dependent, as has been published previously. So we want to understand, well, what happens if you re-challenge these guys with the tumor cells at a later time point? So we perform the experiment. Again, you give the mice 500,000 EL4 cells expressing human CD20. You give them the anti-human CD20 antibody. The mice are survived. The lymphomas is cleared through cytotoxic mechanisms. And then we allowed the mice to rest for 90 days. And then at day 90, we re-challenged the surviving mice with 5 million EL4 human CD20 cells or EL4 wild-type cells. And just important to note that this is a dose that's 10 times higher than the dose we used to prime the mice. And so what you see is if you had naive mice and you give them either EL4 human CD20 or EL4 wild-type cells, none of them survive. But if you take the mice that were primed with tumor and antibody, and give them the EL4 wild type cells, again, you don't see any appreciable survival. But if you take the mice that were primed with tumor and CD20 antibody and give them EL4 human CD20 cells in a re-challenge model, you actually see full protection of these mice, indicating that an anti-tumor immune response in the mice has taken effect, and we term this the vaccinal effect. And it's important to note that this right here, this vaccinal effect, is T-cell dependent, both CD4 and CD8 T-cell dependent, and can be adoptively transferred from mice that were primed with tumors and antibody into naive mice to confer protection. So we wanted to understand what is the mechanism by which the vaccinal effect is generated, and we hypothesized that Fc receptors are involved. And so in order to get at that, we generated conditional mice in which Fc receptors, specifically the activating FcγR4, was specifically deleted on CD11C high dendritic cells. And so using mice in which FcγR4 was floxed and crossed to C11C Cre, you can see that, for example, in the flox control mice, you can see a good FcγR4 expression on your CD11C positive dendritic cells, and then when you cross them to CD11C Cre, you completely lose FcγR4 expression on dendritic cells. By contrast, if you look at spleen resident monocytes, you see a modest but very much incomplete deletion of FcγR4 on these monocytes, indicating that in these mice, you have active FcγR4 on your macrophages and monocytes, but not on your dendritic cells. And so we performed the same experiment before to understand, well, is FcγR4 required on to all cells or on dendritic cells during the primary tumor clearance through cytotoxicity? And as you can see, if you have FcγR4 deficient mice, so this would be the entire immune system is lacking FcγR4, you see a significant impairment in the mice to clear their primary tumors through cytotoxicity, showing that FcγR4 is one of the main effector FcRs during tumor clearance. But if you do the same experiment in the mice lacking FcγR4 specifically on dendritic cells, you can see that the survival curves are exactly the same between R4 flox and R4 flox C11C Cre mice, indicating that FcγR4 on dendritic cells is not required for tumor clearance through cytotoxicity by CD20 antibody. And so what happens then during the vaccinal effect when you re-challenge these mice? So again, the mice are given the tumor cells, given antibody, allowed to rest for 90 days, and then re-challenged with the EL4 human CD20 cells. And you can see again, if you use naive mice, they all succumb to the tumor by 80 days. If you use mice that were given the flox mice, so these are your controlled mice equivalent to wild type, you see a full vaccinal effect. But in the mice lacking Fc-receptor 4 on dendritic cells, you see a significant impairment in the ability of these mice to mount a vaccinal effect, demonstrating that it is Fc-receptor expression on dendritic cells that's required to generate an in vivo vaccinal effect after CD20 antibody treatment of these mice. And so we further characterize this using adoptive transfer studies. Again, we primed the mice with EL4 CD20 tumors, gave them the antibody, allowed them to rest 30 to 60 days, and then adoptively transferred either total splenocytes or CD3 positive cells from the spleens of these primed mice into naive mice, and then the next day gave the mice a challenge of 500,000 EL4 human CD20 cells. And what we see is, again, if you do no transfer or transfer naive cells, you don't see any survival. If you transfer primed cells from the FcγR4 flox mice, the control mice, you see a vaccinal effect that you've adoptively transferred. And then if you transfer mice from the CD11C Cre FcγR4 flox, you can see that you have a significant impairment of the vaccinal effect, again demonstrating that FcγR4 expression by dendritic cells is required to initiate that vaccinal effect in mice. And so at this point, we can summarize what we've seen is you're getting lymphoma cells opsonized by CD20 antibody, engaging Fc receptors on macrophages, leading to tumor cell killing. You're generating immune complexes which are then engaging Fc receptors on dendritic cells, allowing dendritic cell maturation, antigen presentation, and generation of T-cell memory long-term. And so, as I mentioned before, there's significant differences between the mouse and human Fc receptor system. And if you want to study the interactions of human IgG antibodies and the human Fc receptor system, you need a compatible model. And because of this, we generated, I guess I should also point out, there's significant differences between mouse and humans in the Fc receptor system. So mice express the full array of Fc receptors, both activating and inhibitory, on their monocytes and macrophages and their dendritic cells. By contrast, humans express FcγR-3a, one of the activating receptors in humans, on monocytes, macrophages, and NK cells. But by contrast, dendritic cells only express a single activating Fc receptor, FcγR-2a, distinct from FcγR-3a that's expressed on the other cells. And so to understand what's going on in vivo, you would hypothesize that you could generate, through mutations of the Fc domain, an FcγR-3a directed antibody, which, because of the expression of FcγR-3a on NK cells and macrophages, you would be driving more ADCC and phagocytosis and enhancing this process. By contrast, if you generated an FcγR-2a directed antibody, because this molecule is the only activating FcγR expressed on dendritic cells, you would imagine you might be generating more dendritic cell maturation, driving antigen presentation, and creating a more potent vaccinal effect. So to test this hypothesis in vivo, we generated Fcγ R-humanized mice, in which we've made mice that lack all mouse Fc receptors and have been added on transgenes for all the human Fc receptors, so FcγR1, 2a, 2b, 3a, and 3b. And what's fascinating is these mice fully recapitulate the human Fc receptor effector expression and function in vivo. And just to demonstrate, these mice, again, like in human peripheral blood, their dendritic cells are FcγR-2a positive, but FcγR-3a negative. And just as a control, you can see there's some Ly6C low monocytes from the spleen, and they show both FcγR-2a and 3a expression. And so in order to understand which human FcRs are involved in both the ADCC of tumor cells, as well as the generation of a vaccinal effect, what we did was utilize a panel of Fc mutants that selectively engage different Fc receptors. So in these experiments, we have a human IgG1 that has wild-type interactions with FcγR-2a, 2b, and 3a, but then we also have a GA mutant that selectively increases interactions with FcγR-2a. We have an ALIE mutant that selectively increases interactions with FcγR-3a, and then we generated a ghastly mutant, 4 different point mutations that selectively enhance interactions with FcγR-2a and FcγR-3a, but leave FcγR-2b interactions largely normal. And so we performed the in vivo experiment to look at tumor cell killing during that primary cytotoxic stage. And what you can see is, again, we just gave mice 500,000 EL4 human CD20 cells into these Fc R-humanized mice, and then gave them 2 doses of 250 micrograms of human G1 anti-human CD20 antibody on day 1 and day 2. If you give mice no antibody, of course, they all succumb to the tumors by 60 days. If you give them an antibody that selectively enhances interactions with just FcγR-2a, you don't get any increase in survival over just giving the mice PBS alone. But if you give them the antibody that selectively interacts with FcγR-3a, then you get pretty good survival of these mice and tumor cell clearance. And also, if you give the mice the mutant that interacts with FcγR-2a and FcγR-3a, the ghastly mutant, again, you get a very good clearance of the tumor cells through cytotoxicity, suggesting that in vivo, it's FcγR-3a that is mediating the cytotoxicity of antibody-opsonized tumor cells. And just to confirm this with a genetic model, we also did this in mice that express just FcγR-2a, and you can see that if the mice only express FcγR-2a, there's no survival over giving PBS alone. But if the mice express just FcγR-3a and FcγR-3b, you see full survival. So these data collectively indicate to us that FcγR-3a is what's necessary and sufficient for tumor cell clearance through cytotoxic mechanisms in vivo. For sake of time, I don't have time to show the data here, but using clonate liposomes, we actually showed that macrophages, not NK cells, actually mediate the cytotoxicity in vivo. And actually, this was a little surprising to us since FcγR-3a, which is required for clearance here, is actually expressed on the NK cells in these mice. But it seems that even though the NK cells are expressing human FcγR-3a, they still don't mediate cytotoxicity. It is the macrophages in this Fc R-humanized mouse model. And so finally, what about the vaccinal effect? What human FcRs are mediating the vaccinal effect? Well, in order to do this, again, we used our same model, giving the EL4 cells and human IgG1 variants of antibody, allowing the mice to rest, and then re-challenging 90 days later. And what you see is if you give the mice the ghastly mutant, so this is the one that preferentially interacts with FcγR-2a and 3a during that, the mice that were given this antibody and re-challenged show very good vaccinal effect. By contrast, when you re-challenge the mice that were primed with just the ALIE mutant that only interacts with FcγR-3a, then you see that there's a very, very poor and almost completely impaired vaccinal effect, demonstrating that FcγR-2a engagement which is the main FCR, the only FCR expressed on dendritic cells in these mice, is what's required for the vaccinal effect in vivo in the context of the human FCR system. We repeated this in the context of a genetically modified system, and what you see again is if you give the ghastly mutant to fully humanized mice, you get a very good vaccinal effect, but if you do this experiment in mice that just express Fcγ R-3A and 3B and therefore no Fcγ R-2A, you actually get a significantly impaired vaccinal effect there as well, further confirming that Fcγ R-2A expression is required for the vaccinal effect in vivo. And so this brings us back to our model in which we now know that in the context of the human FCR system, it's Fcγ R-3A that's expressed by macrophages that mediates the clearance of the antibody opsonized tumor cells. You get the generation of immune complexes, and they're interacting with a different Fc receptor, Fcγ R-2A, on the surface of dendritic cells. This leads to dendritic cell maturation, antigen presentation, and the generation of long-term vaccinal effect through T cell memory generation. And so why is this important in the larger context of antibody engineering? Well, because a large number of the next generation antibody therapeutics, such as obinutuzumab, are enhanced just for Fcγ R-3A engagement due to the fact that they're afucosylated. Afucosylation largely increases Fcγ R-3A affinity by an antibody, but not Fcγ R-2A. And so we would argue that an ideal antibody therapeutic not only would have enhanced Fcγ R-3A interactions for increased cytotoxic mechanisms, but also enhanced interactions with Fcγ R-2A in order to prime these long-term anti-tumor T cell responses that could provide protection for an extended period of time in the patient. And with that, I wanted to acknowledge everyone in the Ravitch Lab, especially Jeff, and who was an outstanding mentor, and also need to acknowledge our funding sources, Leukemia Lymphoma Society, NIH, and the New York Community Trust. And with that, I'd be happy to take questions.
Injection of the CD20 ER4 cell can also induce the induction of CD20 antibodies, and might that have a role in your protection?
You know, so when we looked at antibody responses in these mice, it was variable, but there was no trend, and most mice didn't actually create a very strong anti-CD20 immune response just using indirect immunofluorescence assays. So we think this is largely T-cell mediated, as shown previously. One interesting thing that we found in the process was it actually looks like this process may be dependent in some part on B cells as well. If you do this experiment in MUMT mice lacking B cells, you don't get the generation of a vaccinal effect. So is that due to antibody production, or is that due to just the splenic architecture being destroyed when you don't have the B cells during the immune system development is unclear at this point, but there could be some role in this process as well for B cells and antibody.
Beautiful study, David. Very clean, very easy to understand in a complicated situation with all these Fcγ receptors. What intrigues me as an immunologist is you bring it down 50%, but there's still that bit. So is that part of the answer?
It could be. So, but it could be in both the human and the mouse. You see that you never bring it down completely. So in the mouse, I think that's easily explained by the dendritic cells and macrophages as well also express Fcγ R-3, which can compensate for the absence of Fcγ R-4. Especially when you lose Fcγ R-4, common gamma chain is still expressed, and you actually get an increase in Fcγ R-3 expression in that case. So I think there's a compensation going on there. But also, even in the human system, when you lose Fcγ R-2A, you still have some vaccinal effect. And I think in that case, you can't rule out that macrophages could certainly be causing this vaccinal effect as well. They're the cells that are mediating a large amount of the ADCC, and that's certainly a possibility. Or it could be contributions from B cells. We know B cells are excellent antigen-presenting cells, especially when they have a BCR specific for the antigen, and they provide important co-stimulatory molecules and MHC class II. So it's certainly possible that it's much more complex than just dendritic cells. I think we're able to clearly show dendritic cells are certainly important, and talk, I think, on Wednesday from the Engelman lab will also show dendritic cells are vital for this type of effect, but I think it's more complicated.
Very nice talk. So I had a question. If you go into the human clinical data, patients that have been treated with CD20 and then genotyping, there's been a lot of papers early on just looking at CD3 or mutants that have higher binding, or patients that have higher binding to Fcγ R-3A, and then other patients have higher binding to by Fcγ R-2A. If you parse the data looking for individuals that are both high binders for 3A and 2A, do you see any increased benefit than the individuals alone?
So in these mice that we used, they all use the low affinity alleles of FcγR2a and 3a.
Yeah, but what about human patients?
But in patients, in the literature, it's certainly clear the polymorphisms, the higher affinity Fc-receptor polymorphism patients seem to do better in follicular lymphoma, I think CLL as well. And they've seen those as well with the HER2 antibodies, and I think even the EGFR antibodies as well. So I don't know if anyone's ever looked to see how their long-term responses or if they get better cellular responses, but it would certainly be interesting to mine that data and see if the patients that have the higher affinity 2A allele, for example, do better when they get the Rituxan treatment over a longer period of time.
I guess the question was if they both have the high affinity allele for FcγR3a and the high affinity allele for FcγR2a, if there's then an improved effect over the individual.
Yeah, that would be a good hypothesis. Sometimes things are a lot more complex in those patients with chemotherapies and things like that, but it certainly is a reasonable hypothesis.
Great talk and very nice paper in Cell.
Thank you.
2 short questions. In the early literature of antibody adjuvant effects, IgM were shown to be very potent in an adjuvant setting, which would suggest complement involvement. Do you have any comments as to whether and what is the role of?
That would be interesting to look. So again, this is all IgG,
Obviously, right.
But it would be interesting to see if you could generate larger immune complexes with the pentameric IgM, if that would stimulate this, and if it would even be a better immunogen than the IgGs. That would be an interesting experiment.
And it could be a complement involvement as well in the system.
Sure.
And also, when you re-challenge, have you looked at higher tumor burden? How well titrated has the tumor burden have to be to see
for these experiments?
Vaccinal effect, yeah.
We've done a couple. We actually increased the tumor burden to get a better separation. So it seems pretty potent. I mean, when we do those adoptive transfer studies, you can adoptively transfer, I think, just about 5 million total CD4 and CD8 T cells into a naive mouse, and you saw about 70% of the mice that you transfer into show protection in that re-challenge. So it's fairly potent.
Great. Thank you.
Great talk. Thank you. So in thinking about the DC role in the vaccinal effect, and you noted that B cells are required, do you have any thoughts on what role antigen spread, whether T or B antigen spread, might be driving this?
So that's something we're starting to look at. In these experiments here, we did not see any epitope spreading. So, for example, I showed you re-challenge the mice with EL4 wild-type cells that don't express human CD20, and you don't see the vaccinal effect. We actually did another experiment. We took a totally different cell line. So we primed the mice with the EL4 CD20 and antibody. We re-challenged them with a different lymphoma cell line, one that expressed human CD20, one that didn't, and we only rejected the human CD20 positive cell line. So in this model, it seems like human CD20 is the only antigen that the immune response is being directed at. But it's certainly conceivable, and something we're looking at now is, if when you're killing these cells, why don't you get other antigens popping up?
Yeah.
Now, maybe the EL4 cells are not super immunogenic, and if you used a different cell line that had more neoantigens that the B6 MHCs picked up, then you might see that. So that's something we're looking at.
Thanks.
Thank you.